Good evening, friends. I'm Rick Myers at the HudsonAlpha Institute for Biotechnology. I'm sitting here in our lab with Dr. Nick Cochran, another one of our faculty members here at HudsonAlpha Institute. And this evening's program is to give you updates about our Memory and Mobility Fund, M&M Fund, or projects, I should say. And these are projects where we are trying to understand and do something about diseases of the nervous system that really are devastating and affect so many people. These are neurodegenerative diseases as well as neuropsychiatric disorders. I will say, just to open, that these disorders and diseases affect just so many people. They're sadly very common. They affect us more as we get older, but they really affect a lot of different ages. I have myself very personal experience with this. My father had Parkinson disease, a neurodegenerative disease. And although my mother succumbed to pancreatic cancer, before she did that, she had onset of Alzheimer's disease. And so this is affecting many, many, many families. And so we put a lot of effort into trying to do something about it. So we're gonna talk to you about several things that we do. And they're really sort of three areas that my laboratory and Nick's laboratory, Nick is now a faculty member and runs his own laboratory, work on. And they have to do with discovering what causes the diseases and what's different about tissues, brain tissues and other things in people who have disease who do not, and versus people who do not. A second area is, can we develop a blood test to detect them? And we'll tell you about that. And then finally, we'll talk about two approaches that we're using to try to do something about it, to see if we can help not develop a cure ourselves, but to contribute to new treatment paradigms that are desperately needed for these disorders. So welcome, and thank you again for joining us. So to talk about what we call the causes, or at least things that contribute to diseases, I'm going to pass over here to Dr. Cochran. Nick, if you could tell us about that.
Sure. So good evening, everyone. So to start to go into some of the details of these three areas that Rick mentioned, the first is discovering causes of these diseases, and that can be in a couple of different categories. One of the categories that we think about is things that you're born with that may confer risk for these different diseases. And in that area, we've been working on this for many, many years, some before I arrived at the Institute, and then I've contributed to many projects at the Institute. And if you've tuned in to these types of events before, you may be familiar with collaborations that we have with, for example, the University of California, San Francisco, the University of California, Santa Barbara, and then through them, the University of Antioquia in Colombia in South America. As well as the University of Alabama at Birmingham, where we've partnered to work on rare early onset dementias. And we've actually worked on rare early onset dementias with all of those groups. Now, those are all things that we continue to work on with those groups, but some new areas that you may not be as familiar with that have been things going on very recently are a couple of initiatives that we've really ramped up and grown based on those existing connections. So one is at UAB with the Alzheimer's Disease Research Center there. So they selected HudsonAlpha as their partner for doing genetic measurements and genetic analysis. And that's a new center that's very important. These Alzheimer's Disease Research Centers are around the country, and this is an important contributor in the Southeast for underrepresented populations, and making sure that we right size the contributions of genetics for those populations. And it's similar to an area that we've grown on research in South America. So we have partnered with 10 different sites across South America from different countries, places all around. So there's Brazil, Argentina, Mexico, others, and we again are the hub center for genetics that is working with all of these centers to help understand both early and late onset dementias for all of those countries. So we're very excited about those ongoing projects that are new efforts in the space of discovery of what can contribute risk for disease. So there are other ways that we can think about discovering causes of diseases. So in addition to things that you're born with, there's the way that your cells and your tissues respond to the environment around you. And there are ways that we can capture and look at that sort of thing in the lab. So I'll pass it back over to Rick to talk about some of some of those approaches in general.
Right. So one thing that is absolutely remarkable is that there's technology. We didn't invent it, but we have it's going in our laboratories where we can take cells from an individual with a disease or an individual without a disease. Usually they can be blood cells, they can be cheek cells that you swab. They often are skin punch cells just from a little piece of skin. Those are not brain cells, those are cells from other parts of the body, but we can reprogram those to become neurons and something called microglia and other types of cells that are in our brains. And that has just made our field move much, much faster because we can now look in the test tube or in these dishes like this. This is a good example of one we grow in what we call our tissue culture or cell culture rooms. They're plastic bottles and other versions of this that look kind of like Petri plates. And believe it or not, we have in the rooms down the hall human neurons growing in these. And we can end up getting millions. I think one of these gets you, 5 or 10 million neurons, or if not more, so that we can then do studies to say, well, what's different about a neuron, a population of neurons in someone with Alzheimer's, ALS, Huntington disease, Parkinson's disease. Those are 4 of the major ones we work on. We also work on something called CBS or CBD, cortical basal syndrome, and FTD, which is frontal temporal dementia. And all of those we can work on in experiments like that and apply technologies that we happen to be very good at, because that's what HudsonAlpha is about, is genomics. We apply genomics technologies to try to understand the differences. And this is advancing our field. We're using that in many different ways, advancing our field very much. So we call these, they're actually reprogrammed cells from other parts of the body that you can then turn into very difficult, certainly difficult to obtain. You don't get biopsies from human brain tissues. So we're able to do that in ways that we couldn't do even 5 years ago. So this has been a real boon for us.
And could we perhaps bring up the picture so that everyone can see what these look like? So, you saw the pink dish of cells, and now if you look at that under a microscope, what you can see are these pictures of neurons, and this is what it would look like under a microscope. You can see that we've done some fluorescent staining where you can see different types of cells. And those are some of the things that we can look at, like, do the proportion of the types of cells change under a disease condition? Do the shape of the cells change? Does the underlying genome change? That's something you wouldn't see visually in this picture, but something that we do look at quite a bit, of course, being working with our focus area in genetics and genomics. So I guess we can go back to the camera now. So another thing that's important beyond what we do in the lab and cell culture and those types of dishes is that we can also, while it's hard to get and it's definitely an important thing to do, to also look at tissue from brains from individuals with these diseases. And that's another approach that we take. There's some very new approaches in the field that are letting us answer some questions that we couldn't answer just a couple of years ago. And this is using the cells from your brain, and you can isolate cells from brains of people who have passed away from these diseases, and then look for differences and what is going on in the genome that may be different, that could help us understand ways that we may either understand the diseases or even think about new treatment approaches. So we've started doing that. We've looked quite a bit already at Alzheimer's disease versus brains without disease. And we've even just recently, so this is data that's just starting to come off of our sequencing machines, looked at Parkinson's disease and ALS as well. So we're very interested in both the commonalities and the differences between these diseases using these very new technologies. So that's another area that you probably haven't heard us talk about before, because it's something very new that we're working on just now in the lab. So with that, I'll talk about another exciting area and pass it back to Rick on early detection approaches.
Before I do that, I want to add something that we've done for years and it's essentially the same project, that type of approach that Nick was mentioning on postmortem brain tissue in neuropsychiatric disorders. These are very, very hard to study. They're hard to define. This has been schizophrenia, bipolar disorder, and major depressive disorder, but the same type of approach. And while it's not the same as working with living cells, it's very, very valuable to be able to do that. So all of these things linked together. When we learn something about ALS, it teaches something about all the other disorders as well, and vice versa. So they overlap a lot, and even the neuropsychiatric disorders do as well. Okay, so Nick mentioned that our second area of focus, that's a really exciting area that we've been doing in neuro disorders only recently. We applied this to cancer a few years ago successfully, so we thought we would try to do this in neurodegenerative disorders in particular. If you could detect that someone is about to get one of these disorders pre-symptomatically, or even early stages of that, or maybe even earlier than pre-symptomatic, very early in the stage, if you could do a blood test, and we like to think about these as blood tests because we actually draw blood, and then we look in the bloodstream with our DNA sequencing and RNA sequencing tools to see, is there something present in people with the disorder versus someone without the disorder? And we've actually applied that in the laboratory with Alzheimer's disease so far. One of the hardest parts of this is collecting the right types of biospecimens, and we really do need lots of them. We need at least 100 or 200 or even 300 or 400 individuals with the disease, as well as matched control individuals, people who do not have it, because we have to do the comparison. But we've done this with 250 samples from Alzheimer's and controls, and sort of really excited about the fact that we have discovered they're not ready for prime time. They're certainly not clinically ready, but we've discovered a set of what we call biomarkers of nucleic acid, or it turns out RNA molecules, that look like they're present much more in people with Alzheimer's disease as opposed to people who don't have it. We need to replicate that on a much larger study, and our dream is to get that so that it's definitive enough that we then find a pharmaceutical company, or maybe another type of large company you can conduct a clinical trial with thousands of individuals, which we do not have the wherewithal or the money to do. This is that we're talking about tens of millions of dollars to do those studies, not to mention that the big companies will be good at doing that kind of work, but working closely with us. So this has been very exciting. We call these our biomarker experiments, but really what we're doing, can we develop a blood test? And the blood test would be good for more than one thing. One of them is early detection. Any disease, anything that you can detect early will have a much better chance of doing something about it in treatment. Even if we don't have treatments yet, like is the case with Alzheimer's, with most of the neuropsychiatric disorders, neurodegenerative disorders, even if we don't have a treatment, these early detection tests can be very useful for the drug companies when they are doing their clinical trials to then monitor whether someone has it early, and also how they're responding to the treatment, meaning as a monitoring throughout the treatment regime. And then once we do have drugs, you might use this test to monitor, as opposed to doing only cognitive tests or movement tests, which are sometimes much harder to measure. So we're very excited about this, have some serious efforts in this. We've done it in Alzheimer's, where we are collecting with Crestwood and some other groups, biospecimens and clinical information from individuals with ALS. That would be a really, really important one, but also with Parkinson's disease and Huntington disease. So stay tuned for that, because we're making rapid progress. Again, we don't want anyone to think that we're ready to start testing them in a clinical setting. We're nowhere close to that yet, but we are making real progress that I'm just delighted to see. So the third area that I'm going to pass on to Nick now to begin this, we're taking two approaches to see if we can do something about treatments. And it's not that we develop drugs per se in our small institute, but we do experiments that help in drug development. And we're very excited about this because there are two approaches that we are, I wouldn't say uniquely, but almost uniquely positioned to make real contributions in. So Nick will tell us about the first.
That's right. So, and the unique positioning comes from history. So Rick's lab for a long time has studied gene regulation and the basic science of gene regulation. And many other labs in the field have contributed to this data as well. So now after many decades, there's a lot of data and some new technologies that allow us to generate things that we couldn't in the past. And we can stand on the shoulders of that data and now take a magnifying glass and look at particular regions of the genome that we're very interested in, for example, because it's a gene associated with a neurodegenerative disease. And we've done this in high detail for a couple of genes so far, and we have others that we're interested in working towards as well. But I'm going to back up before talking about the specifics and say why we would want to do this. So what do I mean by gene regulation? What that means is how a gene is turned on and off. And what that means is that there are ways for the cells in our bodies to control which cells become a liver cell versus a brain cell versus a skin cell. That all is gene regulation. It can also be affected by the environment and things that happen to you over your lifespan. That can all affect gene regulation and perhaps even risk for disease. Now, many neurodegenerative diseases follow a fairly simple mechanism. And what happens is that there are key genes that are either up more than they need to be or down more than they need to be. And over time, what that can lead to is an accumulation of some sort of toxin in the cell, some sort of protein that may aggregate and perhaps could be detrimental to the cells and eventually lead to cell death. There are other occasions where a particular gene may serve as a protective factor. And if it's lost, then cells are more susceptible to neurodegenerative disease. So it can go in both directions. Now, the genes that this applies to that we've made the most progress on, first is Huntington and Rick's lab, and then a project that I've led on a gene called MAPT that codes for tau, which is a gene and protein important for Alzheimer's disease, frontotemporal dementia, and other neurodegenerative diseases. So these are two very important ones that we've made a lot of progress on. And now we're interested in expanding those efforts because we've honed our skills in this area and applying it further to genes associated with ALS and Parkinson's disease. So we're very interested in TBK1, which is a gene that we led the discovery to associate with disease, as well as a gene called Synuclein for Parkinson's disease that we think works very much in this gene regulation mode that we're very well equipped to study. So we're very excited about that and these future directions for this type of approach that we're very well equipped to study based on the history of research at the Institute. So with that, I want to turn it back over to Rick for another type of treatment therapeutic type approach that's brand new that you probably haven't heard about if you tuned into these things before.
Yes. So that what Nick talked about is a way of turning genes, especially off, sometimes turning them on, but turning a toxic gene off. And that's a complicated process, but we have a lot of expertise in understanding how genes get turned on and off, and thinking about how to deliver that for Parkinson's disease or Huntington disease in particular, where it's a deep brain structure, or in Alzheimer's where it's here, and this part of the brain is how do you actually deliver that into? And so that's a hard problem. ALS, maybe a little easier because it's in the spinal cord. So the delivery of components there that would turn those genes off. And our hope is that we, from this knowledge that we're gaining and being able to turn them off, by the way, in the cell culture room in vitro, not obviously in people yet, being able to do that, we really hope will lead to something that can be used therapeutically. And it's not a complete fantasy. People have had some success in a couple of other rare neurodegenerative diseases that we're not working on to actually turn those off. And we're hoping to turn them off permanently, the toxic gene permanently, so it could be a really great way to treat. So that's one approach. And sorry, Nick, you passed it on to me to talk about a new approach. And this really is something that that we began as a collaboration with a very close friend and close colleague in San Francisco, UC San Francisco, Dr. Peter Walter. And Peter and his laboratory, and now probably 100 laboratories around the country and around the world, are studying a process, and don't worry too much about the name of it, but I'll say it is called the integrated stress response. And what this is it's a set of proteins that get together and try to help cells all over our bodies to get back to normal when something goes wrong. Well, guess what something goes really wrong in neurodegenerative diseases. You have cells that are dying and you have inflammation. So this integrated stress response is increased in our neurons and in our brains in trying to deal with that terrible stress that is happening. So that's the process. But the exciting thing is that Peter and his group discovered a small molecule, it's like a drug, that actually inhibits that response. And they've done this in cultured cells and in mouse models of some of these disorders, and the results are very exciting. And we started collaborating with them back in March to do this in human cells, and the same types of technologies, technologies and resources that we have in terms of the cultured cells and disease versus non-disease, so that we can help understand that process. And it almost goes back to biomarkers or early detection again. If we can have a blood test that tells the physicians, hey, this person is now having an increased integrated stress response, then we do the other biomarker stuff to say this might be Alzheimer's or ALS or some of the known diseases. It could be a way of intervening early. And in this case, it would be intervening with the pill that you can take. So we're very excited. It's brand new work. Very excited about this because we're working with a group of really, really first-rate scientists, not just in California, but around the country. And stay tuned for this one too. This is new for us, but we already are doing these experiments and starting to learn things from them as well. So we could go on for a lot longer, I think, in the interest of time, but also in so that we can listen to you. We would love to hear any questions that you might have. And I meant to tell you this at the beginning. I hope it's not too late. If you have questions, there are two ways to do this in a Zoom. One of them is to go to the chat function. If you look at the bottom of your Zoom, you can see chat, and then you can type it in there. But the other is there's a place under in the Zoom that allows you to do Q&A, so you can put questions in there. I suspect people have already been doing this. Many of you may be already pretty savvy at this, and we'll try to answer them as they're read to us. And I'm trying to listen out to see if I can hear those now.
Hey, Rick, can you hear me?
Yes.
Hey, okay, so we have some questions from the audience. So can you clarify where you get your samples to study from, both patients with diagnosed disease and your control samples?
Nick, you want to answer that one? It's a tough question because it's hard to do this.
Yeah. So first of all, for cells and culture, for example, usually these are publicly available resources that are in a repository where they're heavily quality controlled, and those can be distributed to labs across the country. And the cells themselves that we study come from either a skin punch or from a blood draw from an adult, and then those can be reprogrammed into the neurons that we need. Now, the tissue that comes, the brain tissue, so that's an extremely valuable resource. And if you've had a loved one affected with these diseases and been approached about brain donation, it's a sensitive topic because it's something, but it's so valuable for research because we can learn so much. And it's really hard to replace that as a resource. So what we usually will do to obtain those kinds of things, again, these go through very well-controlled repositories. For example, we have obtained a lot of samples from a brain bank run by the National Institutes of Health. And the other thing that's important to note about this is that we will not receive information that would allow us to identify these individuals. So all that we would receive is, for example, their biological sex, their age, their disease status, and perhaps some other technical type of things around the sample. But all of that is meant to protect the people who make this valuable contribution to research by making this donation. So that's the type of thing. And so that's the type of source of samples that will normally happen. It's very similar, for example, for the things that are for sequencing genomes, very similar type of process.
But they're from all over the
They're
place.
from all over,
depending on the study.
Yeah.
So Nick's Colombia, South America, and actually all over Latin America now, South Central and even North Mexico. Those are from clinics that have been working together. It's very coordinated. It requires a lot of samples to do and clinical information to do this. So collaboration with groups like that, it's really hard to do this with one individual place. Several years ago, we did a big genome sequencing study for ALS. That's where we discovered some new genes, some of which are in not clinical trials, but are being developed, we hope, into drug targets. We had something like 3,000 to 4,000 individuals with ALS, and they came from working with collaborators at the other Columbia University, from Canada, from North Carolina, and some from Europe. So that's an important part, and we rarely would do this on a small scale with just our laboratory, because these are such important and valuable resources that need to be spread so that researchers, both can work together, but make discoveries as well.
And I know we probably want to get some other questions. I do want to add one more point about that, and that's that many times we get questions about people locally want to participate, and for the most part we don't do that. There's reasons for that, but there are times that we do when we want to make a local impact. So for example, with UAB, through a very defined mechanism where they're evaluated at a clinic first, and again, we're not seeing the personal information of these individuals, but it's a way for us to make a local impact. For a long time, we've been doing a study where we can sequence the genomes of people with early onset or atypical dementias, and in that way contribute to our local community here. So it's not all just things from national repositories or from these global collaborations. We do deeply care about making an impact locally as well.
We just don't have the wherewithal, at least for these disorders, for people to come in and give samples for us. Now in the clinic, especially for childhood diseases, that's another thing. We have been able to do that. But I will say another local one that's actually just brand new that we've set up is a reset up is with Crestwood for a hospital with ALS. And we're working with them, and they are collecting anyone with ALS and the families, as well as the controls. And the controls are often harder to figure out how to do than people with samples from people with disease. And they not only have, it's a rare disease, sadly, it's not as rare as, there are many people affected, but they also see in their center of excellence there a much larger group of people from all over the Southeast and actually from other places as well. So they are interested in that kind of contribution. We just don't meet them ourselves. We need to have that privacy window or window there to protect the patients and patient rights.
Thank you. So another question we have is, how does HudsonAlpha's work support new treatments and drug development efforts?
Could you say that again? I'm sorry. I didn't.
How does HudsonAlpha's work support new treatments and drug development efforts?
Oh, okay. Shall I start with that one? So that last project that I talked about, and the next to the last one that Nick talked about, is that we're doing research that is trying to figure out, will this be a good treatment? You can also take the very first thing Nick talked about, what causes a disease. When we find genes that are affected in individuals who have a disease that are not present in people without the disease, those genes code for proteins. Those proteins are often targets for drugs, for new drugs, maybe even known drugs, but often for new drugs. So our research, and we don't sit on this for years or months even, when we make those discoveries, we share those data immediately with the outside world, and sure enough, stuff starts to happen with those. And the reason that's important is that we're a relatively small institute. I think we have an outsized impact because we are real experts in genomics and genetics and have collaborations all over the world. But you're never going to do anywhere in an academic setting or in a even biotech setting, something that drug companies have to be the ones to do, because these are the clinical trials. So we feed them knowledge, data, sometimes a lot of knowledge that they don't have otherwise, that then ends up going in that direction. Now, that said, that last project I talked about this integrated stress response, that's closer to the bedside than because the pharmaceutical companies and big biotech companies are already interested in this process. And Peter Walter, my colleague, is already working with some of them to get that stuff into not the marketplace so much, but into the clinical settings as rapidly as possible. And we're part of that. And I mean, I think some of our data will feed into that, like within months or weeks of actually making the discoveries, and how fast that becomes something that's clinically used widely is hard for us to control, but they sure aren't going to do it until we make those kind of discoveries and get it to them.
Yeah, and I'll just add, that generally in academic research and especially in genetics, oftentimes we think about things as target identification that may be useful way down the road. And what we're trying to do is to shorten that way down the road as much as we can, and as much as makes sense in an academic environment. So it still is a long process. And even once it gets to the point where it's an established target, it's a long process for drug companies to develop something and take it to the clinic. But we want to make that process as short as possible. Now, one thing about that, we have a view towards it. And we are here talking about these things that we're actively doing to try and shorten that process. For example, we have the people who think about things this way. So in graduate school, during my first part of graduate school, I worked on an academic drug discovery project. It was a very non-traditional thing for an academic scientist to do. Another key scientist in Rick's lab formerly worked at a pharmaceutical company doing drug development. So we have these people who have thought processes that are geared towards how do we work towards a therapeutic. So,
and that's exactly right. But the other thing that's exciting, this is really why HudsonAlpha formed. The idea was to make discoveries, have the nonprofit research and teaching institute, and now clinical institute, make discoveries, but where all of us have a bent towards seeing something happen with that quickly, including in our own laboratories. But also, I think many of you know, we have 48 biotech companies on our campus in this building and in the other buildings. I think there are 850 or so people. Carter Wells and his team helped to recruit and keep these biotech companies going. And a big part of that for Jim Hudson and Lonnie McMillan's vision was that we would rub elbows with each other, eat lunch with each other, collaborate with each other, even start some of those companies ourselves, so that we can get them into the clinic faster than we could if it was just purely driven by philanthropy and grants, because it just takes too much more to go and turn those into products, if you will, or that kind of application. So that bench to bedside used to be 20 years for anything that we would do. And we at least are cutting off a whole lot of the time that it takes to get to new target molecules for drug companies and the applications, not to mention the biomarkers are clinical, the early detection biomarker blood test experiments are really something that could end up being used much, much more quickly than it would be if we left it to the pharmaceutical companies.
Thank you. So you've talked a lot before about work in Huntington's and more recently Alzheimer's. What is HudsonAlpha now doing related to Parkinson's disease?
Let me start. Nick is doing some on Parkinson's in his lab as well, but I will say I have a long history of this. I mean, it's a little ironic. I started working on Parkinson's disease about 20 plus years ago when I was at Stanford. And we were trying to find genes because there are inherited versions of this. They're rare ones. The irony was about a year after I started, my father was diagnosed with Parkinson's disease. It just happened to be the case. And so again, a deep personal reason. It affects a lot of people, and it's pretty heterogeneous, so it's hard to work on. But so we've done lots of studies over the years, including when we got to HudsonAlpha on these. But these new technologies that you've heard Nick talk about, I'm going to let him take that one up again. These new technologies of working with cells in culture being one of those, and something you didn't even mention yet, but something called single cell technologies are being applied to Parkinson in a big way. So you want to just mention that?
Yeah, so the single cell technologies out of brain tissue is one thing, and that's hot off the press, has just been done. But we have done that now for a handful of samples from Parkinson's disease patients and matched controls. And that data is in analysis right now in the lab. So the other thing, and something that my lab is focusing on, is a key gene that's important for Parkinson's disease. It's also important actually for Lewy body disease, which is related to Parkinson's by this gene, by this protein, synuclein. So synuclein is a gene that aggregates into a protein called a Lewy body. So you can actually, in fact, see Lewy bodies in other dementias besides, or in other neurodegenerative diseases as well. It can be a part of normal aging, but it also has these distinctive pathologies in these diseases. Now, what happens with synuclein, again, is this classic gene dosage model. So just the dose can be too high. What that means, there's rare families where there's an extra copy of synuclein in the genome. And just by that extra copy being present, they absolutely will develop Parkinson's disease if they live long enough. There's also more common forms of Parkinson's disease that are linked in a more risk factor type way with synuclein. So what that means is that the types of things that we do at HudsonAlpha and understanding gene regulation, or how a gene is turned on and off or up and down, is very applicable to a gene like synuclein. So my lab is very interested in beginning to work on that. We're starting to think about that, putting in grants for synuclein. But some of that early work are the types of things that we need to get that preliminary data to be competitive for those grants. So that's the type of early work that we're starting that so critically relies on the generosity of philanthropy that we've received through things like the memory and mobility program. So that's the great type of thing that can allow us to make inroads where we wouldn't have been able to otherwise. So
and we have to stay on the cutting edge and actually help to lead the cutting edge on this. I'm going to expand a little bit, and I hope you'll bear with me. My wife is going to tease me for talking too much on this one, but Nick mentioned single cell technology. When we do over the decades, and I've been doing this and many other people doing it, we will often work, especially if we're working with, let's say, a postmortem brain sample. We might take a small amount of tissue, maybe it's 100 milligrams, a small amount of it. That actually is millions of cells, and it's millions of, not millions, it's many different kinds of cells. So if we take that tissue sample, we grind it up to get the DNA and RNA out, and then we analyze it by sequencing or something, looking at the way the genes are expressed. We're looking at a population of cells, and it's whatever we measure is the average of what's going on in that tissue sample. But what if in those 10 million cells that we grind up, what if about 1% of them are a special type of cell that is particularly changed in, let's say, ALS? We're not going to see that because it's going to get diluted out. So here's I like this because I'm just completely enamored with many of the technologies. There's a machine, I guess the box is about this big, where you put your tissue sample in, and it goes through and takes one cell at a time. Cells are microscopic. You can see them. You saw them a little earlier, but those are microscopic. You can't see those with the naked eye. It takes one cell at a time, puts it in a little bubble so that you can analyze what's going on just in that cell for its DNA and RNA. And then you analyze the next cell. I think we do this for about 20,000, 15,000 cells at a time in one of those experiments. And it's unbelievable how much more we can learn from doing that than looking at it in bulk. Both of them are important, but this single cell technology, we've only been doing it, it's only a few years, 2 or 3 years old, and we've been doing it almost since the beginning of the technology being published. And we bought one of the machines, and it's getting even better and better and all sorts of things. So really excited to be able to learn things from them that way.
Okay, so what does Hudson Alpha bring to the table? Why is what we do important to other researchers and consortiums?
Hear you.
If you want to start,
Okay, let me start and you fill in. So that's a great question because certainly I learned early in my career that the best way to do this is to share data. The Human Genome Project was a great example of doing that, where you don't hoard data, you don't do stuff so that you can be the only one to get credit for it. It's important that you are recognized, that your institution and your laboratories are recognized. That's how we get grants and publish papers and things. But the truth is, we are probably one of the best labs, best groups, I should say, our institute, are one of the best examples that by collaborating, first of all, with many, many groups, but what we bring to the table in the collaboration are unique skills, like some of the technologies, and actually this computer, a lot of it's computing with computers, but this computer too, the expertise. Nick has been working on Alzheimer's disease and related diseases for 10 years, and the knowledge base for that and putting it all together with many, many other people. And that may sound a little hokey that, oh, we collaborate. Well, everybody says they collaborate, and many people do. But I think that it's the culture and a philosophy that the best way to make advances is to share data that way. We started releasing data in the Human Genome Project as data were being generated, as opposed to waiting until publication, for instance. That sped up the way that the data could be used so much faster than it would be if we follow the traditional models. So that's at least one of the aspects. There are many other things. Nick, do you have anything to add?
So one thing that I'll add to that, and one thing that makes us unique in my eyes is that is not even so much what is here, but where it is and what's not here. So the environment. And what I mean by that is that we're extremely focused on applying genomic and genetic technologies to understand biology, and especially to understand diseases. And we all are working on that, where and the expertise here is really, really unmatched. So there's people that have come from all over to come together under this common goal. Even the layout of how we do the research. So all of the equipment that we need is all right here, very close together. The people who have this great expertise sit close together, pass by each other on their way doing experiments. That's very different from what you may see in a university setting, where key pieces of equipment may be spread out between buildings. The people are isolated, often offices in different buildings. And so these incidental interactions that are so valuable in driving the science forward just don't happen as easily. So while they may collaborate with people at their university or externally, it's hindered by these barriers that are in place that are really broken down at Hudson Alpha. And that just exclusive focus, that expertise, that conducive environment really just makes things flow very fast, very easily. And it helps us reach our goals, I think, faster than we could otherwise if we were in a different environment.
And that's a cultural thing that was very, very intentional from our founders, from day one of our founders. But also, we're very proud of our physical plant here, meaning the buildings. There's a lot of glass, a lot of this. You can see everybody, you interact with everybody. We try to have as open an environment as possible. And that actually makes a big difference. I worked in medical schools before, and you had rabbit warrens where you have to walk across campus, and it just made it a lot harder. It's not to say that these other places are weak. They're wonderful. They're great. We collaborate with a lot of them, but we intentionally wanted to make something different, a little different. And another part of that difference is the biotech companies, which help to create an ecosystem of large numbers of people working on life sciences problems. And even if they're not working on ALS or Alzheimer's or neuropsychiatric disorders, we interact a lot with them as well. And that is a unique thing. Nothing else exists like that, I think, anywhere in the world where we're on the same campus with that many different types of groups.
So can you talk more about how discovery in one neurodisease can be translated to another one?
You want to start? You want me to? All right, I talk about this a lot because, so, and Nick knows this well. So at least in neurodegenerative disorders, many of them lead to accumulation of aggregates like the Lewy bodies that Nick mentioned, like plaques and tangles in Alzheimer's disease. Huntington disease has an aggregate that happens in the brain cells that happen, and maybe all of them do, many of them do. This means there's a sort of common thing that happens that might be starting in different parts of the brain for the different disorders, but at least some of the mechanisms are, I was going to say are going to be, we actually know some of the mechanisms are similar. That integrated stress response is an example of maybe anything that happens to these, whichever the disease is, it could end up being very similar. There are differences between the diseases, but what we learn from one truly teaches an enormous amount. But some of that is, we didn't know how neurons die 20 years ago. We really didn't know much about it at all, or even 10 years ago. We're learning more and more. As we learn what happens, you see that there's similarities, as I said, between Parkinson disease and Huntington disease and even and all of them, let's say. And in fact, Nick knows more about this than I do. Some of them are so similar that they often are mixed up, actually. And we give names like FTD and Alzheimer's and things, where what we really want to do is to understand the molecular basis for it, to then distinguish them, but then see where they overlap. That overlap cannot be overemphasized. It's actually one of our strongest tools.
Yeah, and I'll just add to that in addition, there's established spectrums of these diseases. So, for example, ALS is on a molecular and also how it can present in the clinic, how the patients how their symptoms are on a spectrum with frontotemporal dementia. Many patients with ALS will have some central or eventually develop dementia involvement, and vice versa with this disease, frontotemporal dementia. Many will have some aspect of movement involvement. And in addition, for especially for late onset disease, like Rick was saying, many times it's not clear what underlying pathology is contributing to a given disease. And in fact, what's becoming now recognized in the neuropathology field is that it's actually the norm to have a mixture of things that historically we thought were associated with distinct diseases. So there's amyloid beta and tau, and TDP43 and synuclein. All of these things have historically been associated with different diseases, and many times you can see 2 or 3 or all 4 in brain samples from individuals who may look like they have one disease, but in reality, there's contributions from all these different things. So it's really we must study these diseases together to gain an understanding of what's really happening in any individual one.
And that I'm glad you said it that way, because it might seem that we work on too many things for a relatively small institute, but it's because they're so related and we're using the same technologies and approaches to do it that we get a lot of synergy from that.
Mm-hmm.
So I think in the interest of time, we're getting close to the end here. I'd like to say a couple of things. Well, first of all, thank you for your attention, for your questions. If there are other questions that we didn't get to answer, we'll have these written down, and we as you can see, we love to talk about this stuff. And we'll have numerous of these updates, including some guest speakers from out of town occasionally. We've done that in the past, and we're hoping to be able to do that next year. But I want to thank you for all this. I also we want to extend our thanks. Nick mentioned this earlier, that we couldn't do this without the support of the community. I am so blown away with how much support we get from the North Alabama community, Huntsville, Madison, all the ones around us, from the state itself. This is very different from what I experienced in my years in California, where there's a distance between. We feel very close to this community and are greatly appreciative of what you do. And it's not just pay for our whole programs. We write grants, we get other sources of funding, but what we rely on from our philanthropy is being able to do these projects that are too early stage to get an NIH grant, for instance. You cannot write a grant and get it funded if it's at the early stages of some of the ones that we've described. And so this is important and will continue to be important. So thank you so much for all the support that you've given. And we really hope that you will think about, we would love it if any and all of you think about making gifts towards this fund, towards these projects. It's called the M&M Fund, and we will have ways online, et cetera, so that if you're not familiar with it, where you can learn how to give to those. Thank you very much. And thank you, Nick, for all your insights as well.
Thanks, everyone.